Welcome everyone to Life Science Academy online video lecture classes. In this video, we will study and discuss about Agrobacterium mediated gene transfer. So let's move on to brief discussion about Agrobacterium. Agrobacterium comes under phylum Proteobacteria, class Alpha Proteobacteria, order Rhizobiales and family Rhizobiaceae. It is a genus of rod-shaped gram-negative bacteria and it is commonly found in soil. So Agrobacterium is an example of aerobic bacteria and is peritrichous in nature. Agrobacterium has several species and Agrobacterium tumefaciens is the most commonly studied species in this genus. This bacteria is also known as natural genetic engineer because of its ability to integrate its plasmid gene into the plant genome. Agrobacterium tumefaciens infects the plants at their stems and transfer a part of their TI plasmid referred to as tDNA, and this leads to development of tumor in the stem of plant known as crown gall disease, a lump of tumorous cells. Moving on to different species of Agrobacterium. First one, Agrobacterium tumefaciens. Agrobacterium tumefaciens is the most commonly studied species. Its plasmid is called TI plasmid or tumor-inducing plasmid. This plasmid causes crown gall disease on plants. The Agrobacterium tumefaciens infects over different families in dicots, including Leguminosae family. Next one, Agrobacterium rhizosens. It causes hairy root disease in plants, and the plasmid here is called root-inducing plasmid or RI plasmid. It is also known to infect crown gall disease in some plants. The next one is Agrobacterium vitis. Agrobacterium vitis affects in grapevines and it causes crown gall disease on grapevine. The Agrobacterium vitis plasmid is also known as TI plasmid. Next one, Agrobacterium radiobacter. Agrobacterium radiobacter is a less virulent opportunistic pathogen in humans and it rarely causes infection. It is a saprophyte and it depends on dead and decaying matter. Another common species of Agrobacterium is Agrobacterium ruby and it is known to infect cane gall diseases. Agrobacterium uses opines as their carbon and nitrogen source. Opines are arginine-derived low molecular weight compound found in plant crown gall tumors and hairy root tumors. Now moving on to classification of TI plasmids according to the types of opines that are made. There are usually three types of plasmids: nopalin plasmids, octopine plasmids, and agropine plasmids. First one, nopalin plasmids. Nopalin plasmids synthesize nopalin as their opine. These plasmids causes crown gall tumors which can differentiate into shoots with abnormal structure known as teratomas. Second one, octopine plasmids. Octopine plasmids synthesize octopine. These plasmids synthesize undifferentiated tumors and they do not form teratomas. The last one, agropine plasmids. Agropine plasmids synthesize agropine. Agropine tumors do not differentiate and they develop poorly and die early. So this is the structure of TI plasmid. TI plasmid is approximately 200 kb in size. It has several regions like T regions, Verlin's region, transfer region or TRA region, left and right borders at both ends of T region, and an opine catabolism gene region and an origin of replication. We will move on to that lately. Moving on to the TI genes involved in bacteria and plants. Three main sets of genes are present. The first set consists of Vir gene which helps in the transfer of tDNA into plant genome, Shy gene which helps in shoot induction and it codes for cytokinin, Roi gene which helps in root induction and it codes for auxin. The second set consists of opine synthesis and opine catabolism genes. The opine can be nopalin, agropine and octopine based on the type of TI plasmid. NOS means nopalin synthesis gene, NOC means nopalin catabolism gene, and OCS means octopine synthesis gene, and OCC means octopine catabolism genes. The third set of genes are TRA gene which helps in bacterial gene transfer, INC gene which is gene for incompatibility, and an ORIV which is origin of replication for autonomous replication of the TI plasmid. Transformation of plant cells require three types of function in the agrobacterium. The three loci on the agrobacterium, chromosome virulence A, that is CHVA, chromosome virulence B, CHVB, and phosphoglucomutase, that is PSCA, are required for the initial stage of binding the bacterium to the plant cell. They are responsible for synthesizing a polysaccharide on the bacterium cell surface. These three loci are found on agrobacterium chromosome. Next is tDNA. A part of the TI plasmid which is transferred to plant is referred to as tDNA. tDNA is required to transform the plant cell. tDNA contains genes for tumor induction. Size of tDNA is about 23 kilobase pairs. tDNA region contains gene for auxin and cytokinin and opine synthesis. When this TI plasmid is transferred to plant, the action of auxin and cytokinin synthesis in the plant leads to the formation of tumor-like crown gall which is similar to callus of plants. Opines are synthesized by plants and it is found in crown galls. Specific opines produced depends on the types of the TI plasmid but not on the plant host. TI plasmid, the TI DNA is flanked by 25 base pair imperfect direct repeats known as border sequence. It is present at both ends of tDNA known as left and right borders. Next one, the virulence region or Vir region. The Vir region is present outside the tDNA region. The Vir region encodes the function required for the transfer of the tDNA to plant cell. The expression of Vir region is usually repressed under normal conditions, and it only becomes activated when the bacteria senses plant-derived signals from wound sites. This activation leads to the production of Vir proteins, and Vir proteins mediate the transfer of DNA and proteins into host plant cells. Next region is TRA region or transfer region. The proteins needed for conjugal transfer of the entire TI plasmid to recipient bacteria are encoded by TRA genes. Another region is opine catabolism gene. Opine catabolism gene are responsible for the catabolic utilization of opines. These opines can't be utilized by the plant host and will be exported out of plant cell, where it will be taken by agrobacterium cells. The bacteria possess genes in the regions of TI plasmid that allows catabolism of opines. Another region is origin of replication. We know that plasmids are autonomously replicating extra chromosomes, so they have an origin of replication for undergoing replication. Virulence region is a 6 loci consist of Vir A, B, C, D, E and G region located outside the tDNA region. Size of the loci is 40 kilobase pair and some contains more than one open reading frame. The Vir genes are activated by the phenolic compounds released by the plant in response to wounding. These phenolic compounds are released from wounded plant site. For example, in the case of tobacco plant, the generated phenolic compounds are acetosyringone and alpha hydroxyacetosyringone. tDNA in TI plasmid is flanked by 25 base pair imperfect direct repeats known as border sequences. The border sequences are present at both ends of tDNA known as left border sequence and right border sequence according to their position. tDNA has almost identical repeats of 25 base pair at each end in the TI plasmid. The right repeat is necessary for the transfer and integration to the plant genome. tDNA that is integrated in a plant genome has a precise junction that retains 1 to 2 base pair of the right repeat, but the left junction varies and may be up to 100 base pair short of the left repeat. That's why the deletion of the right border can terminate tDNA transfer, but the left border is non-essential because just external to the right border repeat, there's an enhancer portion called overdrive sequence that is required for high efficiency transfer. Moving on to the different types and functions of the virulence genes. Vir-A and Vir-G forms a two-component regulatory system within Agrobacterium. Vir-A is a kinase protein in bacterial membrane and it acts as a receptor for sensing the phenolic compound released by a wounded plant. Vir-A is a histidine sensor kinase protein and will become phosphorylated before passing on this phosphate group to response regulator Vir-G. The activator response regulator Vir-G then binds to Vir box located upstream of each Vir promoter to activate the expression of other Vir regions. Vir-B. It is the largest operon in the Vir region, encoding 11 Vir-B proteins involved in the transfer process of tDNA and bacterial proteins into host plant cells. Vir-B operon synthesize and assemble T pilus, where B4 and B11 codes for ATPases that provide energy for DNA transfer, where B3, B6 and B8 codes for subunits of a putative inner membrane translocase, where B7, B9 and B10 forms a core complex that stabilize the T-pilus channel subunits, where B2 codes for the major pilin subunit of conjugative pilus called pre-propylin, where B1 and B5 codes for minor components of the conjugative pilus. Next one, Vir-C. Vir-C operon encodes for two proteins, Vir-C1 and Vir-C2. These proteins influence pathogenesis of Agrobacterium towards different plant hosts. Mutations in Vir-C can reduce the virulence but cannot remove the virulence permanently. Vir-C1 and Vir-C2 proteins binds to the overdrive sequence present on tDNA and enhance the conjugation. Vir-D. Vir-D operon encodes for four proteins, that is Vir-D1, Vir-D2, Vir-D3 and Vir-D4. Vir-D1 and Vir-D2 are involved in the processing of tDNA. Vir-D1 acts as topoisomerase to unwind the DNA strand, where D2 acts as endonuclease and nicks the tDNA at right border and remain bound to tDNA as it is transferred to plant cell, where D2 also has a nuclear localization signal sequence and remains attached to 5' end of tDNA as a cap. Activity about Vir-D3 is very little known, where D4 serves as a coupling factor that recognizes and transfers the T strand to the transport channel. Vir-E. Vir-E operon encodes for two proteins, Vir-E1 and Vir-E2. Vir-E2 is an effector protein and also single-stranded binding protein translocated together with tDNA into host plant cell. It helps in delivery of tDNA to nucleus of the plant cell because it also contains a nuclear localization signal sequence which marks the protein and the associated DNA for entry into the plant nucleus. Vir-E2 also protects the tDNA from nucleus attack. Vir-E1 is involved in promoting the transfer of the Vir-E2 protein into host plant cell. It binds to the single-stranded DNA binding domain of Vir-E2, therefore preventing the Vir-E2 protein from prematurely binding to the T strand within the bacterial cell. And the last one, Vir-F. Vir-F is a host specificity factor found in some but not all types of Ti plasmid. Octopine type Ti plasmids possess Vir-F but nopalin types do not possess Vir-F. The ability to cause crown gall tumor in certain plant tissue or not has been attributed to the presence or absence of VRF gene. Now let's see about transfer apparatus. Transfer apparatus encoded within Ti plasmid helps in conjugative transfer of the Ti plasmid between bacteria and allow the delivery of tDNA and certain effector proteins into host plant cells. Transfer apparatus is achieved by TRA or TRB system and the VIR-B and VIR-B4 system, which are members of type 4 secretion system. Bacteria has several secretion systems which are protein complexes present on their cell membranes for secretion of substances. These secretion systems are used by pathogenic bacteria to secrete their virulence factor to invade the host cells. Bacteria type 4 secretion system is a bacterial conjugation system by which the protein or protein-protein complex or DNA-protein complex can be transferred directly from the recipient cell through cell membrane. The participating bacteria can be of same or different gram-negative bacteria species. Now coming to the main part, mechanism of infection by Agrobacterium tumefaciens. The wounded plant cells release signaling molecules like acetosyringeone and alpha-hydroxyacetosyringeone. These signal molecules are recognized by dimeric transmembrane receptor complex, virulence A, chromosomal virulence E, that is VIR-A-CHV-E receptor complex. The VIR-A has autokinase activity and it phosphorylates itself on a histidine residue. The VIR-A protein phosphorylates the VIR-G on its aspartate residue. The activated VIR-G now acts as a transcription factor of other VIR genes. The CHVE protein increases VIR-A protein sensitivity to phenolic compounds. Synthesis of attachment proteins. Bacteria synthesize polysaccharides that anchor bacteria to wounded plant cell. Four main genes are involved. First one, chromosomal virulence A, that is CHVA. Second one, chromosomal virulence B, that is CHVB. Third one, phosphoglucomutase, that is PSCA. And fourth one, attachment, ATT. The products of first three gene forms the polysaccharide, and after the production of polysaccharide, a calcium-dependent outer membrane protein called ricadhesin is also produced. It also aids in sticking bacteria to the plant cell wall. The polysaccharide here produced by Agrobacterium is similar to the polysaccharide of symbiotic bacteria. The next step is the recognition of the 25 base pair tDNA borders, that is right border and left border by VIR-D1 and VIR-D2. VIR-C1 recognizes the overdrive enhancer at right tDNA border, endonucleotic cleavage at the bottom strand by VIR-D2, and it remains covalently bound to the 5' end of tDNA complex as cap. Recovery of the T strand of the Ti plasmid occurs. The VIR-B together with VIR-D4 forms a type 4 secretion system channel, and through this channel tDNA along with VIR-C1, VIR-D2 and VIR-E2 proteins enters into the plant cytoplasm. The plants treat type 4 secretion system channel as an infection. In order to escape from defense mechanism, the Agrobacterium synthesize polysaccharides from four genes CHVA, CHVB, PSCA and ATT. The polysaccharide mimic that of compound secreted by symbiotic bacteria, and plant cell then does not consider the channel and attachment as pathogenic event. The tDNA enters the plant cytoplasm through type 4 secretion system channel. The plant cytoplasmic receptor recognizes the nuclear location signals in VIR-E2 and VIR-D2, and nuclear uptake of transfer complex occurs. The tDNA integration into plant genome occurs by an unknown mechanism. Accidentally, in some cases, flagellin in the bacteria's flagella comes under contact with plant's LRR receptor, that is leucine-rich receptor. The plant triggers different defense response such as systemic acquired resistance. The plant triggers the MPKK4 or 5, the mitogen protein kinase 4 or 5 pathway. It leads to the phosphorylation of MPK3, mitogen protein kinase 3. The MPK3 then phosphorylates VIR-E2 interactive protein called VIP1 protein. The phosphorylated VIP1 protein now interacts with Vir-E2 on tDNA. The VIP1 also activates the pathogenesis-related gene called PR gene. Now, the entire complex VIP1 with tDNA passes through the nuclear pore. The system is now hijacked by Agrobacterium. The tDNA enters in nucleus and the phosphorylated VIP1 and Vir-E2 gets dissociated from tDNA. This dissociation is initiated by Vir-F. Vir-F is a ubiquitin ligase, and Vir-F adds ubiquitin tag on Vir-E2 and VIP1, and they are subjected to 26S proteasomal degradation. The free tDNA is now integrated into plant genome via an unknown mechanism. Vir-D2 has an active role in precise integration on T strand in the plant chromosome. The release of Vir-D2 protein may provide the energy containing in its phosphodiester bond at the tyrosine 29 residue with the first nucleotide of T strand, providing the 5' end of the T strand for ligation to the plant DNA. Expression of integrated tDNA genes take place and production of auxin, cytokinins and opines occurs. The opines are secreted by transformed plant cells and is consumed by Agrobacterium for nutrition. The increased level of auxin and cytokinin produce tumor-like growth called crown gall. And so this way the Agrobacterium mediated gene transfer takes place. Thank you.